Today in this lecture, we will discuss four basic principles through which the interstitium of the renal medulla is high in osmolarity. In our previous lecture, we discussed that there are two ways through which kidneys concentrate urine. First of all, there is release of high level of antidiuretic hormone through which there is reabsorption of water from the collecting ducts into the interstitium. And the second thing is that the renal medulla is high in osmolarity. There are more solutes as compared to the solvents in the medulla as compared to the cortical region, as we have discussed previously. Now, in this lecture, we discussed the four basic principles through which the medulla is high in osmolarity. The osmolarity in the renal medulla is high as compared to the cortex. First of all, we discussed previously that there are two types of nephrons: the cortical nephrons which are present in the cortex of the kidney, and the juxtamedullary nephrons which dip down into the medulla. Now, the loop of Henle of these nephrons play a role in the high osmolarity of the renal interstitium of the renal medulla. But along with that, the vasa recta, the blood vessels that dip down with these collecting ducts, they also play a role in maintaining the osmolarity of the high osmolarity of the renal medulla. But the mechanisms, the inside mechanisms that lead to high concentration of solute in the medulla include, first of all, reabsorption of sodium chloride and potassium ion from the ascending limb of the loop of Henle, especially in the juxtamedullary nephrons. Release of reabsorption, active reabsorption, and reabsorption through counter entry from of high number of sodium chloride and potassium ions from the tubule into the interstitium of the renal medulla, from the tubule into the interstitium of the medullary region. Second thing is that there is a reabsorption of a lot of ions from the collecting tubules. From this region, there is a lot of reabsorption of ions from the reabsorption of a lot of ions from these collecting tubules. And finally, there is a reabsorption or passive movement of urea from these collecting tubules into the medullary interstitium. In this way, the osmolarity of the medullary region of the kidney, the renal medullary interstitium, osmolarity of the renal medullary interstitium is very high. It may reach up to 1200 or 1400 milliosmol as compared to the concentration of the 300, which may be in the proximal tubule, and which is the average osmolarity of interstitium everywhere in the body, as well as average osmolarity of all the body fluids in the plasma. Another very important mechanism that lead to high osmolarity of the renal medullary interstitium is no reabsorption of water in this region. No reabsorption of water from these tubules into the renal medullary interstitium, especially in the juxtamedullary nephrons. So reabsorption of sodium chloride and potassium into the renal medullary interstitium, reabsorption of sodium and potassium and other ions from the collecting ducts into the renal medullary interstitium, passive movement of urea from the collecting tubules into the renal medullary interstitium, and finally no reabsorption of water. No reabsorption of water into the renal medullary interstitium lead to high level of ions or solute, and less of water, which leads to high osmolarity of this region, the medullary region, high osmolarity of the medullary region as compared to the cortical region. And this mechanism, these principles are the main mechanisms of countercurrent multiplier mechanism. In the coming lectures, we will discuss each and every step of the countercurrent multiplier in detail. Thanks a lot for watching the video.